This is one of my favorite stories ever. This is a story of how Roche acquired Promedior for $1.4 billion. Promedior was a startup company that commercialized intellectual property developed at Rice University to treat pulmonary fibrosis. Today, we're interviewing Richard Gomer, a former Rice faculty member and one of the inventors of the technology that Promedior commercialized. In this interview, Richard shares how he and his co-inventor first conceived their invention, the process of commercializing it, and the twists and turns that the startup went through to bring this technology to market. If you're interested in commercializing Rice IP, send us an email at ott-licensing@rice.edu or check the description below for more details about Rice IP that is available for licensing. Also, be sure to subscribe to our newsletter to keep up with the latest technologies that are being developed right here at Rice University. Now that that's out the way, let's jump right into the story. You're going to like this one.
So I'll do the whole story of my life. When I was a little kid, I always was interested in physics and astronomy. When I was in high school, I started teaching myself electronics. I went to a place called Pomona College. I was a physics major. I was fully planning to go to graduate school in astrophysics. And while I was at Pomona, I actually started building a detector system to look at light from variable stars that you could mount on a telescope. But the summer between my junior and senior year of college, I got a job doing some electronics for a biology lab. And this is at the University of Chicago. And they need something running at 60,000 volts, which is technically quite challenging. And I'd had a biology class in high school, and we just had to memorize all this crap. And I thought I'd rather stick pins in my eyes than go into biology. It's just so boring and you have to memorize so much crap. So, but I asked these biologists, "Oh, what are you guys doing?" And they said, "Well, kid, you ever wondered how do you get like 5 fingers? Not four, not six, but five." And I thought, hey, that's pretty interesting. How does it work? And they said, we have no idea. And I watched these people working, and they could set up an experiment in the afternoon and let it incubate overnight, and they'd come back and they'd get the results, so they could do things really quickly. They could manipulate the system, and they could do all this stuff all by themselves, and they could do it for relatively cheaply. Whereas when you use a really big telescope, it takes like maybe half a year or more to build the specialized detector equipment. And then on some of the big telescopes I've used, they say, don't be messing around. This telescope costs $1 a second. And when you're doing astronomy, you just have to sit on your hands and just look at the star. You can't change things. You say, oh, it'd be so cool if we could, change the helium content, but you can't. Whereas in biology, you can change things around. So I thought this biology stuff is really cool. So I went to graduate school at Caltech doing biology, but I kept doing some astronomy on the side. Then I was a postdoc at San Diego, and then I started applying for faculty jobs. And I had lots of job interviews. And I really liked Rice because the people there just seemed to be really nice, really welcoming. A lot of places they were kind of a little bit leery of my physics background, whereas in biology department there were people there like Kathy Matthews and Steve Olson who did sort of biophysics, and they were very comfortable with who I was, and I really liked them. So I decided to take a job at Rice. So about a year and a half or so after I started at Rice, I became a Howard Hughes Medical Institute investigator, excuse me. And I had been doing sort of, how do you get 5 fingers, sort of truth and beauty kind of biology. And I was invited to a meeting on sort of size regulation, counting things, basically, in England. And it was at a university where the students were all gone for some holiday. So we got to stay in the student dorms and eat in the student cafeteria. So that made the meeting pretty inexpensive. And I was in the cafeteria, I think it was about lunchtime, and I had this tray of food. And I walked out into the seating area and I looked around and I didn't recognize anybody, not a soul, none of my buddies were there. So there was this one table with a guy sitting there all by himself. And I said, "Well, can I join you?" And he said, "Yeah, yeah, okay." And I started asking him what he did. And he said he was working on arthritis. And I said, "What does arthritis have to do with, counting things?" And he said, "Well, um, if you like are trying to catch a baseball or something, or for him, a cricket ball, and you stub your finger, there are these immune cells called T cells. They come into the joint and they'll work and fix it, and then they leave. But in arthritis, they come in and they never leave. They don't realize that there's too many of them there and they need to go away." So we talked a lot more about the technical details, and I said, "Oh, the T cells might be secreting some sort of factor that they use to smell each other. And maybe in arthritis, they're having a problem smelling each other and realizing that there's too many of them." So I said, "Come to my lab at Rice, and we'll work on T cells." So I was working on this simple organism called dictyostelium at the time. We weren't doing any T cell work, but I said, ask people who I am. Look me up. There are people in England who will say, we do good stuff. So he did that, and he wrote a grant to the British Arthritis Foundation, and they said, "Hey, this is pretty cool. Do it." So he came over with his wife and two children to work on T cells and factors that are being secreted by T cells. And to get the factors being secreted by T cells, what we needed to do then is to take some human T cells and put them into a culture and collect the stuff that they're secreting. Make sense so far?
Yep, I'm with you.
So if I'm going to be trying to purify some teeny tiny amount of stuff that's being secreted by T cells, I want the T cells secreting it into basically distilled water. I don't want to have anything else that I've got to um, try and purify the factor away from. So normally when you culture human white blood cells, or most human cells, or most mammalian cells, you always have a little bit of serum in the culture. And uh, serum has all kinds of stuff in it, all kinds of proteins and small molecules. It's just all kinds of stuff. So that would mean we'd be trying to find a needle in a haystack. And I'd rather be trying to find a needle on a, absolutely clean linoleum floor. So I said, what we've got to do is put the T cells into absolutely serum free medium. So we did that. He thought the cells were going to like die or explode, or the world was going to come to an end. But um, he came to the lab with his family. He already had a place to live. He started experiments about a day or two later. And so maybe 3, 4, 5 days after coming to the United States to work on a T cell secreted factor project, he came down to my office and says, "Hey, you got to come and look at what I'm seeing in the microscope." So when you take human white blood cells and you culture them in serum, what you get is these, little bitty round cells, really boring looking. They're tiny, they're round, and with these cultures of the human white blood cells in serum-free medium, we're seeing a few, little bitty round cells, but there are a bunch of these incredibly long, skinny cells in the culture. So if a normal white blood cell is like this, these things were huge. And we realized that these were a cell type called a fibrocyte that is associated with wound healing. So a long time ago, around 1850, there was a guy named Paget who was looking at wounds. He might have been making them on his own skin or in mice or rabbits, just little skin wounds. And he watched white blood cells coming in, and he could see through a microscope. He was doing drawings with a pencil that the white blood cells would actually form big, long, skinny cells. And what the long, skinny cells are trying to do is pull the wound closed.
Mm.
And so these fibrocytes that are derived from human white blood cells, these are um, really important for wound healing. But there's this whole class of diseases called fibrosing diseases where you get scar tissue in an internal organ. So this is things like pulmonary fibrosis, cirrhosis of the liver, end stage kidney disease, and basically heart failure. And collectively, these fibrosing diseases, these scar tissue diseases, kill more people than cancer. And you also see fibrocytes in these diseases. So, from the experiment with plus serum, little bitty round cells, no serum, big, long, skinny cells, what that means is that there's something in serum that prevents the formation of these big, long, skinny cells, the fibrocytes. With me so far?
Yep, I got you.
So, if these long, skinny cells are involved in these horrible, fatal diseases, these fibrosing diseases, and there's something in serum that prevents it, that means there might be something in serum that inhibits fibrocyte differentiation. So um, we got pretty excited. We almost had to go home and change our underpants, but um, it was like the world, just looking in the microscope, the world just fell out in front of us because we realized that we might be onto something, could be a major therapeutic just from what we saw in the microscope. So we got a bottle of human serum. We dropped that whole T cell project and we've never gone back to it. And we started fractionating serum to find out what in serum is inhibiting this fibrocyte differentiation. And we tracked it down to a protein that's made by the liver, secreted into the blood. It's called SAP. And we can
You said sap,
purify.
like tree sap? Like, how
It's
do
an
you spell it, I guess?
it's an abbreviation for a protein called serum amyloid P.
Okay, gotcha.
The abbreviation is SAP, serum
Okay.
amyloid P. So I'm going to be calling it SAP.
All right.
And so we used SAP purified different ways, and later we could make recombinant SAP, and we could show that yes, indeed, this strongly, potently inhibits the formation of these long, skinny cells. Then we learned how to do animal models like mouse models, rat models of, for instance, pulmonary fibrosis. And we could show that injections of SAP could inhibit pulmonary fibrosis. And if you had rats where you caused pulmonary fibrosis and waited until there was a little bit of fibrosis, and then you start injecting the SAP protein into the rats, just intravenous injections, you can make the fibrosis actually go away. So
Wow.
not just prevent it, but you could reverse it. So this was really exciting. We showed that this works in other systems, working with a guy named Mark Entman over at Baylor, showed that it worked in a uh, a mouse cardiac fibrosis model. But after we got the pulmonary fibrosis, um, initial pulmonary fibrosis results, a buddy of mine named Larry Kovar, who I had helped him start a company a long time ago, was in town. We were having dinner, and I told him about this stuff, and he said, "Whoa, this is really cool. I've got a little company. I'd like to license it in." So we worked with a tech transfer guy at Rice named Peter Schurman to set up a license deal between Rice Tech Transfer, and Larry's company. And eventually, instead of it being a subset of Larry's company, some of the venture capital people who had been funding Larry's company said, let's just have a standalone company that will just focus
Okay.
on fibrosis. And that was uh, naming that company was uh, quite a challenge. So You can edit this out if you want, but the basic idea was me, girlfriend, daughter, all the venture capital people, Daryl, would all come up with ideas for the name of this company. We'd
Hmm.
send them in. And there's always, every time that somebody had a problem with the name. So, like, I like to collect rocks. I thought, oh, Jasper would be a good name. One of the venture capital people, his first girlfriend, her name was Jasper. The memories were far too painful, couldn't be Jasper. So,
Okay.
um, we could not come up with a name for a company. And eventually we had to pay a branding company $35,000 to come up with a name. They came with a name, Melior, Medior.
Okay.
And we had found a CEO, a guy who would be the CEO of the company, and he set up a little office in Malvern, Pennsylvania. Uh, and he printed up letterhead and business cards and got the name Medior registered as a corporation in Delaware. And right after he got all this stuff done, I got a website for Medior. He got a letter from some lawyers, and the letter said, "Sir, you're at 1400 Schmort Street in Malvern, Pennsylvania, with the name of a company, Medior. Well, we're the
Hmm.
Melior Corporation, and we're at 1800 Schmort Street. So if you have Medior and Melior on the same street in the same town, and the address ends in 00, and the first digit of the address is 1, all the packages, all the FedEx, the mail is going to get all bollocksed up. Dude, we were here first. You got to change the name of your company. So it went
Mm.
from Medior to Promedior. So that, at that point, the company, I would go to Boston and New York and do my little song and dance about, the science and what was going on to venture capital people. And I got them interested. So we got a consortium of venture capital people, in addition to the first VC guy, to fund Promedior. And
And then real quick, at this point, are you just serving as like an advisor to the company? Like, are
yeah,
you an equity stakeholder? Okay.
I'm an advisor with equity.
Okay. Gotcha.
So when we set everything up, I got some equity, Daryl got some equity, and of course Rice got some equity, and then the deal included royalty. I mean, scale of royalty payments, yearly milestones for this, that, and the other. Uh,
Yeah.
for um, if you start a phase 1 clinical trial, you have to pay this milestone. Phase 2, got to pay this milestone, et cetera, and so forth. But Rice also got equity.
Okay. And then before you go back to the investor part, I'm just curious, how did you guys find that first CEO? Like, was that something that your the guy you had dinner with, had already had like a recommendation, or did the VCs provide the CEO for
Oh,
Medior?
okay. So the buddy of mine who I had dinner with, his company was being funded by uh, partially funded by a VC,
Okay.
and that the VC who was funding his company was uh, the guy who said, you know what, we need to do. So all the VCs funding my buddy's company said, yes, let's license in this technology from Rice. But
Okay.
then as we started getting more data showing this stuff really had legs, the SAP protein story really had legs. Uh, One of the VCs said, you know what, rather than this being a subset of this one company, let's form a second company that's just completely focused on the fibrosis and SAP stuff. And so that the management of the company, rather than trying to deal with two different projects, can be focused. The last thing you want in a company is the management having to spread their efforts around multiple disparate projects. And so that VC and some of his VC friends, when you're one of these top VCs, you have a list
Mhm.
of all kinds of people who would be good to be a CEO, CFO, and then for the CSO, that's very much dependent on what the technology is. So, for instance, if you were a VC, you would have a file drawer with names and details on all kinds of potential CEOs and then you would say, this one here, she would be really good as the CEO for this company. And she's either unemployed or you call her up and say, hey, Susie, uh, I really need you to be CEO of this company. I'll pay you, $100,000 more per year than what you're making at the company you're at. So that's how it works. So uh, the VC sent candidates out to Houston to come to
Mhm.
Rice to meet with me and Daryl. And there's this one guy, and we just fell in love with him. He had management chops. He was really nice. He was really understanding. He understood the science. He was just everything we wanted. And so we told the VC, oh, this is the guy we would really like to work with. And he became the CEO. He happened to be living in Malvern, Pennsylvania. And so that's why the company started in Malvern. And the
Okay.
company started out for most of its life was pretty close to a virtual company. So what the company then did with the VC money was they needed to make the SAP protein as a recombinant protein. So what first thing he did was he went around to all kinds of contract research organizations that he knew of and said, "Hey, can you make this recombinant protein? I'll give you $50,000." And so went to all kinds of different companies. And one company had a technology that was able to successfully make the protein. I think they sent some of it to us for us to test in the lab at Rice. And at that point, they had a source of the protein, and it was a source in which they could manufacture it in a way that it would be FDA approvable. It's called good manufacturing practice, or GMP, or
Okay.
pharmaceutical grade recombinant protein. And then the next thing that this virtual company did was um, they now had hired science people, and they would go around to all kinds of labs that were really good at studying, say, liver fibrosis or kidney fibrosis or radiation induced fibrosis, and say, hey, we'll give you $50,000 if you test some of this compound for us. And they would work out how they would do it, what exactly the experiments would be. And so because of that, again, not a whole lot of work needed to be done in Malvern, Pennsylvania, because they were having the testing done at labs all over the country. And then the next thing the company had to do was test things like how stable is this protein, how toxic is this protein. So there are companies in the US that you send them a potential drug, and they will very carefully test for potential toxicity with this assay or this assay. So again, all the Malvern operation needed was a freezer to store the protein, and then they would FedEx it to these people to test this way and these people to test it that way. And everything worked out really well. Um, I was then serving as basically just an advisor. I talked to the Promedior people once a week, and trying to do, further research on SAP. At one point, I went through a really gruesome divorce, and there's two Rice faculty, Janet Bram and Mike Guston in particular, who really, really helped me through this very difficult time in my life. I'm deeply
Mm.
indebted to them. And things worked out pretty well for Promedior. Um, they raised a total of 76 million in venture capital funds,
Okay.
and
Wow.
and the interesting thing is a large pharmaceutical company came to them and said, "Hey, we'd like to uh, put in an option to buy Promedior if your phase two trials work." Okay, because they had gotten phase one safety trials to work. And they said, we'll put down 150 million as an option to buy it. And but the interesting thing is they were so sure that they were either going to exercise the option and buy the company for, I believe it was going to be $1.2 billion dollars, or it was going to be a complete failure and they would just have to walk away from the 150 million, that they never bothered to set things up to get equity in Promedior. They never thought that there could be something other than total success and them buying the company and total failure and just having to throw the money away. So,
Wow. Okay. So I
okay.
guess if I chime in real quick, so that's a really big decision. Like,
Yes.
because 100 an option is essentially you're just reserving. It's kind of like you're putting it on hold, like you're putting down a deposit.
Yes.
But $150 million, option just to have the exclusive right to buy it. I've
Yes.
never heard of an option fee that high, and then not getting anything exchanged. So
That was
I kind of know
really
what
dumb.
happens, but I'm curious to hear in your words how it's played out.
So, okay. So then what happened is that first company, after putting down the 150 million option, just wrote a check out the door without getting any equity. Uh, the senior management started realizing, oh, poop, we've got all these drugs that are, blockbusters that are making huge amounts of money, and they're all going off patent in a year or two. We hadn't really been thinking about this. And in a year or two, we don't have anything in the pipeline that's going to be a patented drug so we can sell it for huge amounts of money. We're in deep doo doo. We need to have something that we're selling that's bringing in revenue. And there was another pharma company that had a bunch of stuff that was making money. The patents had still had 10, 15 years life on the patent. And they went to the company and said, we want to buy you how much? And they haggled and haggled, and they came to the, very reasonable sum of, I believe it was something like on the order of $70 billion.
Yeah, just a little
Uh,
cheap
not too.
chunk
So
change.
at this point, the first biotech company who had put down the option now had to scrape together, all, from this bank account and this bank account, looking in the couch, 70 plus $1 billion. So at that point, they could not be doing any additional deals. So they just shut down every deal that they had in the pipeline, which included the option to buy Promedior. So when they
Mm,
shut that down, they walked away from their 150 million option. Now, 150 million is, sort of peanuts compared to, coughing up 70 billion to acquire a big pharma company. So it wasn't, it was a little blip on the finance books. But anyway, so Promedior
mm,
basically got 150 million for free, and they used that money to do really, really successful phase two trials.
okay, great.
The SAP protein was working really well in patients with pulmonary fibrosis, and then a big pharma company called Roche came along and said, "Hey, we'll buy you for in a $1.39 billion deal." So the company was at this point, after that option, worth 1.39 billion. So the original option was to buy the company for 1.2 billion. So
Mhm.
they would have 3.5. So they would basically save themselves 40 million with that option. But in any case, Roche bought the company. I got a nice big check because I had equity in Promedior. My buddy Daryl, he got money. Rice got money, and this is in addition to like yearly milestones. and various other milestone payments that Promedior had been paying to Rice. So the money came in different tranches, but one of the larger tranches, rather than doing a direct deposit into my checking account, I said, I want a check. I want just a hard copy
Physical
check,
check.
a physical check. And they said, no, okay, we don't usually do physical checks for these kind of sums. So, but I got in a FedEx the check. So I put on my grubby T-shirt and my grubby pants, and I went down to the Bank of America, um, and went up to the teller and said, I want to deposit this check in my account. She says, yeah, okay, kid. And she picks up the check and she gets her stuff. I said, oh, I'm not allowed to deal with a check like this. This is more than I'm allowed to deal with. So she, how the tellers have that little ding ding bell at
Yeah.
their teller
Call a
station.
manager,
Yeah,
somebody
ding
from the
ding, so
back.
ding ding ding, and the floor manager comes over and says, "Deposit authorization." He says, "Yeah, sure." And he whips out his pen and he looks at the check and he goes, "Oh, I'm not allowed to deal with a check like that." So they said, "You come back here and uh, talk to the branch manager." I'm sure they had called the police, okay, and we're like checking this, seeing if this check seemed to be legit because
Yeah.
And after a
Had to be.
while, the branch manager comes in. He's wearing a suit and a tie and really nice shoes. And he sits down, he's holding the check and he's looking at me. He looks at me and he looks at the check and he looks at me and looks at the check. He says, what's this from? And I say, drugs. And then he does this little dance in his chair. And he almost went right over. But then I told him a very abbreviated version of the story I just told you. And so it went through. I should have filmed it. It was pretty funny. And
Uh,
so that's my story. Uh, I moved. I After this horrible divorce that Janet Bram and Mike Gustin helped me, move through.
mhm.
They're such incredibly nice people. They're such unbelievably nice people at Rice. That's the huge plus of being at Rice. Such incredibly kind and thoughtful people. But anyway, about a year after that divorce, I was asked by somebody up at Texas A&M to help teach a class where they're teaching graduate students all the different organisms that people use for research. I worked on this organism called Dictyostelium. So little soil amoeba crawls around, eating bacteria on soils, but you can use it for all kinds of different stuff. And so I said, yeah, sure. Well, it turns out he was co-teaching the class with a new assistant professor who had just joined the department. And she was single, and I was single, and I met her, and there was some reception for some heavy duty speaker. The night before, I was going to be giving my teaching this one little class, and so I came up the evening before and I met her and we talked and talked and talked and I just fell in love and went home and I was staying with this guy who was co-teaching the class and I said, "Who is that girl I was talking to? I'm going to marry her." And that's actually what happened. So I moved up to A&M in 2010, but uh, I deeply miss Rice. As you probably know, there's just constant chaos here at Texas A&M. We just lost our president, so Rice was such a nice place to work at. But anyway,
Yeah.
I'm up here at Texas A&M.
No, that's a great story, Richard. I'm glad you shared that. Like, this is my second time hearing it all, but I still have a lot of questions for you, and
Okay,
it
shoot.
always brings a smile to me. Hey, so I guess real quick, like when timeline wise, so you moved up to A&M in 2010. Like when did the acquisition happen where Roche actually, bought Promedior?
That was in the announcement was in 2019, and then all the paperwork was dealt with. I mean, there's huge teams of lawyers to
Yeah.
consummate the deal in 2020. So between 2010 and 2020, I was just had my fingers crossed about Promedior, hoping somebody was going to buy them and something was going to happen. But in the meantime, I've been developing new uh, next generation therapeutics, working on this dixtelium stuff, and working on therapeutics for a completely different lung disease. That, again, do you remember how I said that we had been doing just pure truth and beauty biology? And then
Yeah.
Daryl came into my office and said, "Hey, you got to come look at this in the microscope." So, sort
Hmm.
of something very, very similar happened here at A&M. We had been working with um, proteins being secreted by these dixtelium cells, and a graduate student um, came into my office and said, "Hey, you got to come look at something in the microscope." So very similar. And these mutant cells were basically not moving away from colonies. And from this, we realized that this secreted protein was being used by colonies of cells as a repellent to sort of get cells at the edge of the colony to move away from the colony.
Mm.
And then another really amazing graduate student found that there's a human equivalent of this repellent protein. And there's this horrible disease called acute respiratory distress syndrome, where you breathe in smoke from a house fire, you aspirate vomit, and there are these immune
Mm.
cells called neutrophils come into the lungs, and rather than helping, they start causing a lot of damage, which causes more neutrophils to come in, and this will kill the patient. And if you could just push the neutrophils out of the lungs, this could be potentially a therapeutic. And we had found this repellent protein with dixtelium. We found a human equivalent of the repellent protein. It works as a repellent for neutrophils. So we're now trying to develop therapeutics for this acute respiratory distress syndrome. So everything, all of this stuff is just based on just completely chance observations, people being, really observant and telling me, "Hey, I'm seeing something weird," and then off we go in a new direction. It's an incredible amount of fun.
Amazing, amazing. Look, you're on to part two of the of your career journey. That's exciting. Hey,
Oh yeah.
so I guess so 2010 you moved to Texas A&M. Around 2019, 2020 the sale actually happens. Let's back up just for a quick moment. When do you recall actually filing the patents on this, like at Rice
Oh,
University or
at Rice
that
with
first discovery?
so Daryl came to the lab in 2001.
Okay.
Uh, we had this, poo our pants epiphany moment looking in the microscope. 4 days later, 5 days later, it took us about a year to figure out that this SAP protein was the factor in serum that was doing it, and
Hmm.
maybe another half year to show that injections of SAP protein would inhibit pulmonary fibrosis in rats. And then we immediately started filing patents, working with
Okay.
Peter Sherman at the Tech Transfer Office.
Gotcha, gotcha. Man, that's exciting. So throughout this journey, I'm curious, like, were you ever tempted to like maybe leave Rice and actually join the startup like full time as like chief scientific officer or anything like that?
Oh
Like, I'm just trying to get
no,
to
no.
never. What made you like what helped you make that decision? Like, hey, I want to just stay on this side of Rice and serving as an advisor versus like jumping headfirst into like entrepreneurship.
Um, well, let's see. It would be like me asking you, have you ever decided to go to the airport and start flying uh, um, 747 airplanes? Just this afternoon, because you don't know how to do that. I don't know how to do this CSO stuff. So once you've gotten the basic discovery, then the interesting thing about how these biotech companies work is you need all kinds of expertise that a regular schmo like me or you absolutely don't have. So, for instance, you need to know exactly what kind of paperwork you need for to to file investigational new drug application. You need to know exactly what sort of work you need to have good approval for toxicity and safety. So these areas of expertise are so specialized that the makeup of the company keeps changing. So I have a picture somewhere of all the people at the company. It was taken, and there are all these people standing around the desk smiling. And 6 months later, all but three of them were gone. And so
Wow.
in biotech, you have these people that I call butterfly people. They're really good at some very specific thing. It's like, you know how you have your uh, your expertise, everything
Yeah.
you know. Well, and I have my expertise, and I have everything I know. So these people, they know a lot of stuff. Nobody else knows it, and they butterfly into a company, and they do this very specialized thing, maybe the toxicity assay, supervising the toxicity assay. There's all this stuff with supervising the manufacturing. So you have to make absolutely sure the manufacturer is doing the manufacturing just right. So when you do, um, supposing I was going to try and manufacture a drug in this office, okay?
Hmm.
This would have to be a GMP facility to manufacture the drug. For a GMP facility manufacturing a drug, there are 10 pages of regulations about exactly how the sealing is made. What kind of sealing tile, how the sealing tile is sealed, how the lights are sealed, to make absolutely sure how it's cleaned. 10 pages, just the sealing. Okay.
Just the
So
ceiling. Oh man.
just the ceiling. Okay. So there's just enormous amount of expertise needed in all these different aspects of drug development. I don't know any of it. So I would have just been like a paperweight at the company.
Gotcha.
Once we had found the original stuff, then they need to bring in experts, all these experts. And the other thing that companies will have is some expert who knows which experts to bring in. So she has no idea how you do this or how you do this, but she knows that you need somebody who knows how to do this, and she knows who to get. And so the butterfly people, they're lining up their gigs like, a year, 2 years in advance. And they're brought in by the expert.
Hmm.
They come in, they butterfly in, they do their stuff, and then they butterfly off to the next company that needs them. So um,
Gotcha.
short explanation, I wouldn't have been any use.
Gotcha. Hey, so did you also see that similar pattern happening with like on the executive level, even like the CEO, or was that first original CEO that was a really good guy? Did he kind of stay along for the entire ride?
Oh, no. Oh, that's a really smart question. No. So um, uh, we straight off told this guy, we love you, we think you're just the most amazing dude. And he said, well, boys,
Yeah.
here's how it works. I'm a really good CEO for starting from nothing. Like, I know how to like rent an office, get the electricity and the phone connected, get letterhead and um, business cards made, get the company registered in Delaware, kind of get things moving with um, manufacturing the drug, testing the drug, and then they're going to fire me, because then they're going to bring in a CEO who's really, really good at mid-stage company stuff. And that's exactly what happened. They let this guy go. He was really, really unhappy, but he knew this was going to happen. And they bring in a mid-stage CEO who's a specialist on supervising early stage clinical trials. You've got to like make absolutely sure, you know, everything is being done just right. And then he also was able to supervise the next stage clinical trial. So you've got to make sure the the manufacturer is manufacturing the drug you need and sending it out, and all the different places that are testing the drug on patient are doing the right thing and are filling out the right paperwork and testing the patients in the right way. And then he got fired because they needed now a CEO who is really going to be really good at trying to do a deal with pharma companies and either sell the company or do an IPO. So remember I was telling you about Butterfly? People
Yep.
technically, there's the same sort of thing, but slightly longer time scale for even for upper management.
No, that's really helpful because I think somebody, whether it's at Rice or somewhere else, they are involved with the technology and maybe they're trying to make a decision of, hey, do I want to take a shot at being CSO? But then also like understanding like the business life cycle, like what happens, what changes take place in leadership within the company. So I think that your experience helps give them some valuable insights there.
Yeah, absolutely. So if you go into biotech, um, you're probably going to be at a different company in a few years. There are a few jobs where um, you have some sort of job stability. So there was one person out of all these different people at Promedior. What he did was he would take batches of the drugs that had been manufactured and test them for purity. Make sure making they'd make 1,000 vials of the drug. He'd get one of those vials and make absolutely sure it was the right thing before they sent the vials out to the hospitals that was testing this on patients. And but just about everybody else in for a while,
In and
out and
out.
then gone. Yeah, but
Man. Okay.
the cool thing is this is what makes American biotech really strong because in the American biotech industry, um, there's nobody thinks less of you for being at a company that failed. Like
Mm,
you're at a company, it was working on something, everything was looking good, and then all of a sudden, you know, everything works on rats and works on pigs and dogs, and then you start giving it to people, and they start getting terrible rash. Close the company down, shut her down, and in other cultures like Europe, it's considered, well, you're a failure because you're associated with a failure company. That's to a first approximation. Whereas in the US, it's thought that, well, you've learned stuff, you know, and you go from company to company and you learn more and more and more. And the more you learn and the more contacts you've made and friends you've made, the more valuable you are. But there's
gotcha.
generally little stability.
Man, okay. No, that's really helpful. It seems like the VCs, like the friend that you, it's a few different chance encounters. One is meeting Daryl at the conference.
Yeah.
That was like really the catalyst to spark all of this, and you guys having this unique collaboration where you were able to kind of make both parts of the pieces of the puzzle fit. But then the other one I think is like this dinner with your friend that started a company, and if he didn't come into the picture and if he didn't have the VCs, I don't necessarily know if this happens. Like maybe you end up filing a patent on it just kind of out of,
Right.
happenstance, but it seems like that's another key part of your success. Do you feel like anybody that's going into the biotech realm or entrepreneurship, they should early on start talking to VCs and trying to see, hey, what the potential is or what potential investors may be interested in this type of idea?
Oh
Or what
yeah.
do you
Oh.
kind of think there?
Oh yeah, yeah, yeah. I That's a really, really important point you brought up. Thank you for bringing that up. For all of you out there in podcast land who are thinking of going into biotech or any other business, start um, making a list of all the people you know and make efforts to meet people. So I go to various science meetings, and several times now I've been asked to teach a little thing on how you go from a discovery in a regular lab to commercialization. And so I look out into the room and I say, all of you, you're coming to this meeting, and you've probably come to this meeting with, maybe you're a graduate student and you've come with two other graduate students from your lab plus the professor. And you're probably all sitting together there at breakfast, and you're all sitting together there at lunch, and all sitting together at dinner because new people are scary. They might bite you or say something rude. Yeah, you're stupid. Um, and So what you uh, need to do is get out of that shy mode and go to sit with people you've never met. Don't be sitting with all of your friends, and don't be hanging out with your friends at the whole meeting, as I see this happen a lot of times. Just go find some table at lunch or breakfast or dinner, and there's a bunch of strange, scary people, and sit down and say, "Hi, who are you? What do you do?" And then take notes on who it was you met, and keep those notes, because maybe 10 years from now you say, "Oh, I was at this meeting, and uh, I need to work with somebody who does transflabulation. As an expert on transflabulation, I met such a person. And then you can call them, hey, remember we had lunch? Da, 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 da. So it's really, incredibly important to make lots and lots and lots and lots of friends. So most scientists are really shy, and so this is a hard thing for them.
Yeah, no, I think that's a huge one. And as an engineer and self-described introvert, I feel like that's something I had to learn over time. Podcasting surprisingly helped me, get out of that
Mhm.
shell and be more comfortable talking to strangers. But
Yes,
to that point, like nobody can be successful just working in their own silo at their own desk. Like you have to
right.
find the right people to collaborate. And
Yes.
that's what I love about your story the most.
Yeah. So, yeah. Oh, absolutely. Make lots and lots and lots of contacts, and then keep a record of these contacts. So, it's useless if you say, "Yeah, I remember 10 years ago I was at some meeting. I was talking to somebody. I don't even remember what they looked like." And then it's like, so you actually have to write this stuff down, keep records of it, and
Yep, the
maintain this
documentation.
big network of contacts. Yeah, good
Yeah.
point.
Looking back on this entire story and journey, is there anything in particular that you felt now that you look back where in hindsight it was a mistake, or maybe if you would have done something differently, things could have turned out even better for you? What's kind of your thoughts there?
I don't think we made any mistakes. We ran into Along the way. We ran into some. Uh, you know how there's sharks in the ocean? So
Yep.
there's sharks. There's sharks out there, and ran into some sharks, and some of the sharks, kind of took a little bite. Um, so, but that's just part of the thing. If you're in a situation where there's really nothing you can do, or somebody, you know, um, basically steals something, there's not a whole lot you can do. So I don't think I made any mistakes. I think we were actually quite lucky. Um, I was just unbelievably good luck to be at Rice, where surrounded by such nice, kind, helpful people. There's a lot of people who are work in at universities where you're sort of siloed, and you kind of have to
Mm.
for some reason you just kind of keep working on this, and you just work on that. And Rice and the HHMI gave me the freedom to try this and try that, and you know play around. So I think it was just good luck, one end to the other.
You always crack me up when you tell me the part of the story about you going to the bank and getting the check, or
Yeah,
number one, requesting the check from Roche from the acquisition, but then taking
yeah.
it to the bank. Honestly, I think I would have done the exact same thing
Oh
because,
yeah, yeah,
like, when else in your life are you gonna see a check with however many zeros or whatever?
yeah. A lot.
But I'm just curious, like, was it an I don't want you to tell a number, but was it enough for you to think about, retiring and not, working anymore? And if so, like, what kind of made you decide to still continue this like journey of creating and solving problems and all that?
Oh yeah, I could have retired quite nicely on that check for sure. Um, the there's a lot of studies and a lot of personal observations from me and my friends that that a lot of men, not so much women. Women tend to have more balanced lives. They have their friends and their families and hobbies and stuff. But men, for a lot of men, if you plot a pie diagram of their interests, other than sex, there's their job, and then everything else is this tiny little slice. And so that when they retire, you may have even seen this personally yourself. They'll retire and they just fall apart mentally within that first year, and they're often dead within that first year because they don't have
Mm,
anything to do. There's nothing to um, get up in the morning. And the wonderful thing about the stuff I do as a research professor is I, I wake up in the morning and I'm just so excited to go into the lab. And sometimes I'm doing little experiments, or mostly it's other people doing little experiments. And I can't wait to see what they found. We've got all these little irons in the fire, all kinds of little things, oh, is, Susie gotten back to me? Has Billy gotten back to me, on this? And, trying to do little deals or little collaborations. And it's just exciting. So I'm going to just keep working until either I keel over or uh, I'm I have to teach, just like at Rice. I teach
mhm.
up here at A&M. I actually teach very similar material, and if I get to a point where I can't stand up in a classroom and teach, then I go work in somebody else's lab and say, hey, can I have a lab bench? Give me a little project and sit there doing stuff. But I love what I do. It's just the funnest thing ever.
But I have seen that where, retirement, and you have a great analogy there, or you have it mapped out exactly right. But what I wanted to say though, the other thing I like about your story is that you benefited from both sides, right? And this is something that working in tech transfer, when we're talking to faculty members, PIs, even students, if they don't take the full, head first jump into entrepreneurship, like being a co-founder of some sort, typically we do see them be involved as like an advisor that receives an equity stake, and getting them to understand like, hey, you benefit from the revenue sharing side on the university side, like you get a piece of the royalties, the profits and all that stuff that comes in from the university side. But then also if the company is acquired or IPO'd or whatever, and there's a liquidity event, you benefit from that side as well. So that's the other part that like you kind of like double dipping in a sense. And I think that's a
Right.
great way to,
So
benefit from the work you've done.
I'm going to give you the other side of the coin, which is what happens when you don't work with a tech transfer office.
Okay.
So this is for all of you people out there in podcast land. Listen to me carefully. Um, so the SAP protein is something made by the liver, secreted into the blood, and it inhibits fibrosis. So the people at Promedior had been just, keeping an eye on the literature and somehow came across a paper showing that there was another protein secreted by the liver into the blood, completely different protein that looked really good as a potential therapeutic for a completely different disease. So they, they they called up this. They thought, we're making a recombinant protein, liver blood protein. We could try, on the back burner, have a back burner project, have this other protein. So we've all the expertise for manufacturing proteins and testing them, right? We know how to do this, so it would be really easy to add this on to the technical stream. So they called up the people of this university, the researchers, and said, it's sort of like dating, like the first thing you say is, "Are you seeing anybody?" So I said, "We're interested, in what you got." And the people said, "Oh yeah, well that's really cool." Then the Promedior people said, "Well, are you working with anybody else? Are you, dating? Are you married? I don't want your husband shooting me." And uh, they said no. And so the next question, of course, is, well, what's your IP position? What do you have in terms of patents? And so this was stuff that had been published in a
Hmm.
in a journal. It was right out there. And oh, these people said, well, we figured if anybody got interested, we'd file a patent.
Ugh.
Uh, okay. So what the Promedior people had to do was say to the people at this university, well, for all of the patients with that disease that this protein might have been cured, might have been a cure for, it would have been better for all those patients if your parents had never met. If when you were little, you got run over by a truck, or if you had decided to become, a locomotive engineer or something like that, because you have just completely screwed it up for those patients. No biotech company in the world will ever go near this protein because there's no patent protection on it. And so, um, everybody out there in podcast land, if you discover something, keep your mouth shut. Don't publish it. Don't talk about it at a meeting. Go talk to this guy right here. Tell him what you found. He'll get you a lawyer. He will, um, make sure the patent gets filed. And as soon as the patent is filed, then you can tell everybody about it. So um, that's really, really important. So not only do you make money, but you can avoid doing something that ruins the chances for patients. And if that protein had been the only thing that could have ever cured that disease, then these people, by publishing it, not talking tech transfer, then wrote a death sentence for all the people with that disease.
Man. No, that's somber dose of reality in terms of
Yeah,
what it is. Because I
yeah,
know the researchers, they're doing it because they want to help people. But the corporations, to your point, like the amount of millions of dollars you have to invest to commercialize this, they want to make sure that, hey, any and everybody can't, just take this off the shelf and go do it if we're going to be investing that much money in it. So yeah, we tell all faculty members, engage with us early and often. Don't wait. But at the same time, we work with their timeline if they do have a conference coming up and it's hopefully in a month or two. We can at least still assess the technology, get with our counsel to get something filed. There's even been some extreme cases. We don't try to make this a norm, but there's been some extreme cases where they got a week. But yeah, no. So you were saying you've heard of what?
oh, I've heard of situations where uh, somebody says, "Oh, I'm going to, I've got to give a talk at 10 o'clock tomorrow morning at this big meeting on all this cool therapeutic we've discovered." And somebody says, "Did you patent it?" And there's no. And there's a call at, like, 4 o'clock in the afternoon to tech transfer, and tech transfer scrambles. And you know how tech transfer has this list of attorneys with different skills, and an attorney
Yeah,
can charge quite a lot of money to stay up all night writing a patent and then filing it. And the attorney will do that. They just stay up all night and then they file it at, you know 8 a.m.
yeah. And
So that's
in those situations,
extreme and
we
shitty.
they Yeah, and is I probably had one similar to that, not that extreme, but these big law firms, they'll even take advantage of, their national companies sometimes global companies. And so let's say you're at a university in Maine or in New York on the East Coast, it's 4 o'clock your time. That patent agent, that patent attorney will draft it, and then maybe they'll, work with somebody within their company that's in California on Pacific time, so they have more daylight, more time to develop it and get it at a good point where you can submit it. But then also, filing something is better than nothing at all. So we can get
Yes,
something filed
yes.
and take whatever draft of the presentation or manuscript you have, write out some claims for it and get that filed off. That at least like plants the flag and says, hey, um, this is our first public disclosure date, and we're saying we're the first ones to do this according to effective this date. and then from there you have a year to massage it, develop it
Yes.
further, and submit a non-provisional.
Yes. And so for all of you people out there in podcast land, um, Andrew will have a list of attorneys with all kinds of different specialties. So when we were working at Rice with Peter Sherman, he said, "Well, I'm going to, hook you up with a patent attorney." And this is the first time I'd ever done a patent, so I thought we were going to be talking to some schmo who did divorces and drunk driving, and have no idea what the hell we're talking about.
Jim
So
Adler.
so I thought this is going to be unpleasant. And so Peter hooks us up with attorney. It's Michelle Coint at Baker Botts, and she says,
Okay.
"Okay, I looked at your stuff, and do you know anything about the receptor? Does this involve the NF kappa B pathway?" And it's like, "Oh, whoa, what's your background?" So the stuff we were doing was with um, immune system cells, and a particular. So the immune system has different parts of the immune system, and we were working on this part over here of the immune system, and she was an expert on that part of the immune system. And her story is kind of interesting. She grew up in a very rural area of West Virginia. Right.
Moonshine country.
Moonshine country. I used to make jokes that she didn't wear she never wore shoes until she went to college, but that's not actually quite true. But um, she uh, she was really smart, really smart. So she went to Harvard for undergraduate education. So I guess her first day at Harvard, she walked into the lab of somebody who is working on. Remember, I was talking. Was it this part of the immune
Yeah.
system? This part of the immune system that we're doing stuff on. And she went into his office and said, "Hi, I'm Michelle. I'm a new freshman here at Harvard, and I've been thinking about IgE and mast cells, and which is what you're working on. And I've been, looking at your papers, and I think, what's probably happening is, yada yada, yada connects to the, I don't know, blah blah blah." And she said he just looked at her. He said, "Well, young lady, you're completely wrong, but I've never ever heard of a Harvard freshman, you know, knowing this much stuff about what I work on and thinking like this. I will pay you if you come work in my lab." And she said, "Well, okay." So she worked in the lab of this big cheese immunologist for 4 years. So she has this incredibly strong background. So, all of you guys out in podcast land, Andrew has this list of attorneys who are like experts on whatever it is your patent is on. So, when you go talk to Andrew, and say, "Hey, we've discovered this, yada, yada, yada," he'll be able to connect you with somebody who's, technically really savvy. So do it.
Yeah. Yeah, no, that's a great point. I'm glad you made that as well because we have a number of law firms that we work with, and then they have a number of patent agents, patent attorneys that a lot of times, to your point, like we can just match them up. They'll look at, hey, what was your thesis? What was your research focus? What's your experience? And we do a pretty good job of matching it and finding the right agent that can write the most comprehensive application for the inventors. So I'm glad you mentioned that. Look, man, I need to get you need to be the mascot for the office, man. I need to get you out
Okay.
here talking to all these
Okay.
people. You make my
Okay.
job easy.
Okay. That's what I want to do. I want to, if there's anything where maybe from this podcast that one person who is thinking, "Oh, I've got this discovery, but I won't bother patenting it," um, goes talks to you and does do something, this would thoroughly be worth it.
Yeah, absolutely. Richard, man, this was a lot of fun, man.
Yeah,
Like,
yeah, thoroughly.
look, like you when you get this, I don't know what's gonna come of this new technology, but hopefully it really is a big success, not only academically, but hopefully even gets commercialized and licensed and all that. I know it's not rice technology, but look, I'll have you come back on the show and talk about it, and we
Okie
can
dokie.
hear
Yeah,
how that
thoroughly.
story went
Yeah, it'd be fun. Okay.
for sure.
Bye, Andrew.
Yeah. Take care, Richard. Thanks for
Uh,
joining.